Hi everyone, welcome to another edition of Metaflow Office Hours. Today we have with us Eddie from Outerbounds, who's going to present about Geneformer. It sounds like a super interesting topic. And just to introduce Eddie, Eddie's background has been in scientific computing and then more recently building machine learning developer tools. He has worked as a researcher in academia, in customer-facing roles, and also as an engineer at previous MLOps startups. He was also a product manager at Intel. Currently, Eddie is working to improve the open source Metaflow project and is building tools for AI researchers and MLOps developers at Outerbounds. Eddie, this sounds like a great and very interesting topic. Over to you.
Thanks, Sri. Let me go ahead and screen share, and then we can dive right in.
Sure.
Feel free to interrupt as we go as well. I'm hoping you can, especially with a smaller group on the live call. I'm hoping we can kind of have more of a conversation around these topics. But my game plan is to start with sort of why are we studying Geneformer, computational biology in general at Outerbounds. Sort of move into what we actually did after that, and then final just kind of talk more about the Metaflow piece, maybe a little bit more relevant to people watching this who are more familiar with Metaflow. So just to kick things off, we wrote a whole blog post about this. It's a pretty long read, but we collaborated with Nick Wisniewski, who's a computational biologist. He's kind of been in that space for decades. Very, very familiar with sort of all the complexities of scientists doing what we would label as the purple hat research, like the data science, kind of the scientific computing workflows, and then how now computational biology workflows are trending much more towards the sort of structure of AI workloads, much like the ML engineering space has encountered over the last 5 or 6 years. So we kind of collaborated on understanding what's the landscape in that realm, of what's actually changing in how computational biologists are getting their jobs done, what's related to the different things that AI researchers are doing now, and then sort of what's the state of how do they actually get their job done? What's the infrastructure commonality between what AI engineers do and what these computational biologists do? The upshot of it really is that transformers are very hot in computational biology, much like they are in language modeling. And as we'll see later with the Geneformer case study that we did, we'll use Metaflows to kind of orchestrate the fine-tuning of many different versions of this Geneformer model, which is actually a HuggingFace transformer model, much like the open, if you're going to use open source transformers these days, that's the package everyone uses. So we'll see it's very much the same exact code. It's using a BERT model that was originally made for language data to model DNA or RNA sequence data, to be more precise. So I'll just give a really high level blitz through the post. This table is kind of the lit review that we did. So just pointing out that since August 2021, there's been a lot of activity. Here we see the Geneformer example came in March 2022, and actually there's a really wonderful Nature paper describing all the details. If you want to get into that, definitely a great read. Even if you don't have much biology knowledge and you know a lot about AI, it really goes deep into kind of the use case, and you can bootstrap the bio knowledge quite quickly, I think. There's been a ton of activity recently. There's this Evo model has been making quite a bit of noise in sort of the RNA sequence modeling community. But the point of showing this table is that it's a long list, like several nature papers scattered throughout here. They all have open source code. They're all based on transformers, and these are just biomodels that are modeling RNA sequence data. So this isn't even like the whole AlphaFold paradigm as well, which is another wing of computational biology where people are modeling protein sequences. So this is just to illustrate the depth and kind of emerging sophistication of using these transformer techniques in biology.
And quick question, are all of these kind of sort of based on BERT? So fine-tuned on BERT, or some of them are kind of sort of foundational in nature?
Good question. In the sense that all transformers are sort of based on BERT.
Sure.
You could say that there's like a common lineage there. But there's a lot of innovation happening in these models as well. It's not just like they're taking, like it would be like it'd be very easy, I guess, to kind of misinterpret what I said in the
Yeah.
introduction here, that they're just stealing things from the language community and not innovating on the transformers themselves. That's false,
Hmm.
because these people are making very, very like impactful kind of model architecture design choices around how to make better transformers in general.
Got it. Okay. Sounds good.
Um, Yeah, especially and one quite interesting example there is the hyena DNA one, which is all about running very efficiently.
Hmm.
So yeah, it's like kind of taking the same workload, getting the same performance, running it 100 times more efficient.
Hmm.
Run it on a couple GPUs instead of hundreds of GPUs. These kind of
I see.
arguments, there's a lot of innovation like this happening there as well. Another kind of schematic I wanted to show that was sort of one of the impulses for us being interested in this at Outerbounds is we've been focusing a lot on how to provide access to compute to our customers. And what this was is just kind of showing that the largest model at a couple time points, you can sort of see the trajectory is somewhat like in with the language models, where we see the language models in green. So on the bottom left of the axis, we have BERT, 340 million parameters, and we can kind of see the exponential scale up to GPT-4, which is the current known largest model. Obviously, this isn't public, but it's pretty well known from people scouring the internet that it's about 2 trillion parameters. Um, Unclear if this will continue in the biology models, but the Evo model recently is 7B. It's as big as kind of the Llama 2 smallest model in terms of parameter count. So the point here is that very similar computational requirements are kind of evolving in the biology space that have a bit of a lag, but mirror the NLP space. Okay, so that's kind of then we can just kind of get into like why is Metaflow useful? This is sort of more for biologists who are unfamiliar with Metaflow and MLOps in general for the rest of the post. So what did we actually do though? We did kind of along with the literature review, we sort of used a bunch of these different models. And the one that I want to focus on today is this Metaflow Geneformer example, where Geneformer is a pre-trained model. It has about 100 million parameters. And one of the really cool parts of this paper as well is they also open sourced the data set that they trained on, as well as a really meticulous description of how they kind of guaranteed the quality of the data in all the different sources that it came from. So the data set is called Genecorpus. It's also on HuggingFace, and it has, I believe, 30 million examples. Around this is where the Genecorpus 30M comes from. And these are all different, essentially like DNA sequence or RNA sequence data with really rich annotations around kind of what part of the cell did it come from, or what gene does this RNA sequence belong to. So that's kind of the pre-training mechanism, and it's much like how the GPT models are just kind of trained on a big dump of text from the internet, and they're sort of just learning the basic autoregression task of predict the next token. The same thing is happening here with these big piles of DNA and RNA sequences. The transformer is learning in the pre-training phase to just predict the next token, and it's kind of using this attention mechanism that's kind of common to all the transformer models to understand the dependencies, ideally the long range dependencies in these sequences, which you can kind of, it's a bit of an extrapolation, but you can kind of imagine like how this might be useful in looking at RNA sequences to kind of look across long range interdependencies there. So the idea is there's kind of new emergent things that these models can do that previous techniques that were common in bioinformatics cannot. Now, did you have a question, Shreya? I saw you.
No. I was just nodding along. I was like, yes, this makes
Okay,
sense.
cool. Cool. Now, what we're doing here is we're actually, so another wonderful part of the HuggingFace repo is that the authors prepared a bunch of examples. So we have downstream fine-tuning tasks, one where given a new set of cell data, which they don't really show you too much. So this was kind of part of what we had to study and understand on our side. It's like, how do you take the kind of files, the data structures that you would already have as a biologist and kind of bring your own data into this world? If you're curious, it's all based on this thing called loom files, .loom, which is sort of an abstraction just on top of the HDF5.
I see.
It's very common in scientific computing. Anyways, so you get these matrices that have cells on one axis and genes on the other, and then kind of a bunch of different like membership data is what the actual like NumPy array like object is. But anyways, you can bring these files in, and then there's all of these different kinds of downstream tasks that you can do. So they show you how to kind of annotate these Loom files to prepare them for fine-tuning the Geneformer to do cell classification based on strings of these RNA data, or strings of the tokens of the RNA data, to be precise. Similarly, there's you can classify by gene. One really interesting use case that they highlight a lot in the paper is in silico perturbation, which is very common technique and increasingly highly used by drug discovery companies, where what you're doing is you're kind of using the sort of latent knowledge space that the pre-trained model has gained. And you're kind of understanding, okay, if I can wiggle around these different parts of the genome, say if I could have a drug that would target that part of the genome or this like RNA sequence, what kind of effect could we imagine is become like, how can we cross over the threshold from say being a disease or not in this organ for these cells? So the idea is kind of this systems biology approach within silico perturbation of sort of connect these kind of token level interactions happening on the RNA sequence to higher level properties that we might care about that are more relevant from a healthcare perspective, or from a drug discovery perspective. So anyways, back to the code. What the Metaflow piece looks like is we took the cell classification one in this example, and in the utils file, we have kind of the normal sort of data science code. We have loading data from HuggingFace, caching it in S3, things like this, like lots of Metaflow code that people on this call, watching this call, have probably seen. And then similarly, we have the model ops, which I'll go into in a second. So you can see what I was saying earlier. It's like just a BERT model from HuggingFace. Interesting bits in the workflow. We're essentially going to fine tune N models, where N is the number of different organs that are in the training data set. So we're going to fine tune a different model for each organ that we have represented in the training data. And then the idea is to be able to classify for each of these different organs, is it in a disease state or not?
And sorry for like absolute basic question here. Organ is literally like a human organ. Eyes, nose, ears, hands, like whatever.
Brain, hearts, kidney, this kind of thing.
Got it. Okay.
Yeah, I can show, I think there's some printout that we can see in the UI in a sec that kind of shows the basic gist of that there. But yeah, you can see like we're just kind of running a basic data split, like very basic Python kind of data science code here. We run the for each split, and then we're going to be training on GPUs using Kubernetes, custom Docker image to install the Geneformer. It's in a public ECR registry if you want to play around with this stuff. So the code should run as is for you. And then one of the parts I want to focus on is we're using the dynamic cards to kind of construct our experiment tracking dashboard, sort of like a tensor board light kind of experience that hopefully we can keep iterating on and bring up to speed there. So the actual fine tuning code is going to be in the utils file. And what I'm going to focus on is just showing that it's actually the BERT model, and then showing how do we actually construct these cards that we'll see in a second in the Metaflow UI. So I'll keep scrolling through. This is kind of more pre-processing code to split the organs, or to split by the organs. The actual fine-tuning step we have here, again, basic transformers library. The Geneformer code is wrapped up into its own Python package. I'll come back to this callback in a second, because this is kind of where the HuggingFace magic is that will produce the cards that update dynamically. But yes, this is the piece that I wanted to kind of prove all the stuff I've been saying. It's BERT for sequence classification, very kind of vanilla HuggingFace model, maybe more than 5 years old at this point. So in terms of transformers, it's an ancient model, but it's really kind of a standard NLP tool that people are using. Yeah, and then it's just use the HuggingFace trainer as you normally would. And the important part is this callback. So I'll pop back up to the callback here. And we can see that we're using a bunch of these different dynamic card features. So like we'll see that there's kind of the markdown pieces that have always existed in Metaflow. Now we're also going to be constructing these tables. that is where we're going to see kind of the real-time updates, where we have a progress bar that is tracking the HuggingFace epochs and the HuggingFace steps, as well as some of the loss curves and the metrics that HuggingFace spits out as part of these hooks in the trainer lifecycle. So let's go over to the UI and see what's happening. So it takes a while to run, like some of the longer models take about 30 minutes to do the fine tuning on the data set size that I'm using here. So I did it kind of like the cooking show demo, where I can trigger one actually and show you what that looks like. So if you're not on Argo, this might be a new piece for you. This is the Outerbounds version of the Metaflow UI, but somewhat similar. One feature that I don't, Sri, correct me if I'm wrong, but I don't think this exists on the open source side, is you can actually just click a button to trigger the Argo workflow on Outerbounds.
That's right.
But this is the same as like if you did Argo workflows trigger on the CLI that I'm doing here. This path is just, there's a couple of different sizes of the Geneformer model. So I'm just telling it to pull a kind of generic baseline one here with this argument. But when I click that, I should be able to pop over here and see another workflow started in a second, or at least one queued up on the Argo side. Let's click into the one that's already running now before I do that. So you can see there's, I think, 1, 2, 3, 4, 5, 6, 7, 8, 9 organs that we're training on. A different model for each one. Again, trying to kind of take that pre-trained model and make a classifier for whether or not the RNA sequences that we're seeing in this organ is diseased or not.
So again, like super basic question here. So this was a cell classification notebook that we saw earlier. So is the high level question right now for this model is, given a cell, identify which organ this cell belongs to? Is that
great
a
question.
fair assessment or no?
No, so that's actually already part of the dataset annotation that I'm using here.
Yeah.
So in the pre-processing, it's more like I already knew which cell each of these RNA strands belong to.
Correct.
And what I want to do now is classify, like the labels that I'm using for the classification task is the state, the disease state. Either it is a disease or it's not a disease, essentially is the simplest way to describe it.
I see. Okay.
It's a bit more complicated than that, but like the high level question is knowing that this RNA strand comes from this type of cell, is it diseased or not?
Got it. And then because diseases can be per organ, having one model per organ is one logical way to think about it. That given an RNA sequence for, let's say, the brain, is this RNA sequence a disease or not? Given an RNA sequence for, say, heart, is this RNA sequence a disease or not? And that RNA sequence for heart, especially, again, I'm probably like simplifying it like 17,000 orders of magnitude, but that's kind of sort of the goal, that for a given organ, there is a data set which has RNA sequences, and I mean, the training data includes RNA sequences. And now we are fine-tuning this model for detecting whether given RNA sequence is a disease or not.
That's exactly right. Yeah.
I see.
So again,
to
your point though, it's a design choice, right? You might imagine that maybe it's actually better to just pool all of them together and kind of keep this. Maybe there's something about the latent space of doing like kind of continuing more like the pre-training in that way, where everything is just lumped together. This was part of the author suggests this, and I think as you described, it makes sense to kind of think about different diseases might appear in different. So let's actually look into one of these tasks that's still running. So you can see it spitting out the HuggingFace standard out. We also have the GPU utilization cards here. So we're using V100s, just one per job here. Pretty solid utilization. So probably not a huge cause for concern there. And then we see this is kind of what I was showing earlier. I'll make this a bit bigger. The real-time cards are kind of showing us that we're nearly finished here. So this must be epoch 9 of 10, almost at the end of the progress bar here. And we have kind of a little, here's the parameters that we use. So as you tune these in the config.py file back in the GitHub repository, these will all kind of get logged in a dashboard view as well as save just artifacts of the Metaflow in the Metaflow artifact store. That looks like the grad norm isn't getting locked. But anyways, we can see the learning rate that we're using here. So it's actually a linear decay. Is how the learning rate is being scheduled throughout the job. The train loss and the evaluation loss. So we see kind of clear evidence of overfitting in this example by looking at these two. Our train loss is going down, but after a step about 5,000, we see kind of skyrocketing evaluation loss. So kind of a lot of room for hyperparameter tuning improvements here. And then at the end of the step, so I'll go to one of these ones that's completed. At the end of the step, it's also kind of plugging some nice information that HuggingFace collects for us that shows kind of like the overall runtime and things like the total number of flops spent on the GPU. So you can kind of get a sense of like what's the flops per second here, which I actually don't know if this is good or not for the V100s, but it's quite low compared to many of the modern deep learning expectations I have.
I had a question. So the GPU card, is that just a built-in thing for Metaflow that you called, or is that like a custom card that you implemented?
Yeah, so the GPU utilization is built in Metaflow profilers. Shri, again, can you remind me, is this open source as well already?
It is open source. Yeah, it is
Okay.
open source. You can use GPU profile, even the source code. I mean, and if you want to tweak it or something, you can always take the GPU profile and add some of your own metrics that you want. But yeah, it is available in the open source community. I mean, in open source.
Awesome, thanks.
Yeah, we used to do the custom card a lot for this as we were developing it, but now it's Metaflow.profilers. The API should be the same though. You just set the interval every 1 second, kind of look at the NVIDIA SMI output is what's happening.
Gotcha, yep.
Cool. Yes, that's pretty much it for my demo. Shri, did you have any other questions or?